chapter 7.4 is going to deal with how photosynthesis has evolved over billions of years, really. I'm going to start out with a little bit of like an evolution story time. So we're going to relate back to when the Earth first formed about 4.5 billion years ago. For the first billion years on Earth, there was no life. It wasn't until about 3.5 billion years ago where life first evolved. Now, life that or the first life that evolved was anaerobic. This type of life actually will die in the presence of oxygen. There were low levels, almost no oxygen on Earth at that time. Atmosphere was made up of mostly nitrogen, carbon dioxide, and methane. So life that first evolved was anaerobic, which means it dies in the presence of oxygen. And I know it doesn't seem like, oh, how could that happen? Yes, life can die. Certain bacteria can die if it's exposed to oxygen. So again, because there was no oxygen on Earth, the first life on Earth evolved to live in the presence of no oxygen. So about 3.4 billion years ago, so a couple hundred thousand years, couple hundred million years after that, I should say, the cyanobacteria started to evolve photosynthetic processes. Now, this wasn't per se exactly like the photosynthetic process we see today in plants, but it's very, very close, and it's kind of like the humble beginnings of photosynthesis. This was a huge evolutionary advantage. They could take carbon out of the air and they could produce sugar, which then could produce a lot of usable energy later on. So again, we see a huge evolutionary advantage in this biochemical pathway. You can see the cyanobacteria here. The first life on Earth was long filaments of bacteria cells. And you can see here we have this red sulfury looking layer of the Earth. This is showing us the oxidation that's going on at that time, and the red, kind of like the sulfury locations in the crust that tell us that oxygen was being produced at this time. Now, crazy as it may seem, about 2.5 billion years ago, there was so much oxygen being produced that we're actually going to see it's called the Great Oxidation Catastrophe, or the Great Oxidation Event. This is because most of the anaerobic life at that period started to die off, and it's because there was so much oxygen in the air. Photosynthesis kind of ran out of control at this point. It was a greenhouse gas. There was a great decrease in the CO2 because, again, we have all these photosynthetic bacteria living now. We have less CO2. When there's less CO2, it gets colder. When there's more CO2, it gets hotter. And then we're seeing that today in today's climate change. When there's less CO2, you're going to get colder temperatures. And about 300 million years ago, there was this huge ice age, and there's a couple ice ages that killed off a lot of the life on Earth. But we see a bunch of these ice ages and the large amounts of oxygen in the atmosphere having just destructive events on these anaerobic bacteria. About 2 billion years ago, we see our ozone forming. Again, this is about a billion and a half years after photosynthesis occurs or starts to evolve. This gives us protection from UV light. So you can imagine our Earth before that point didn't have much of an ozone, didn't have much of an atmosphere. The plants allow a ozone to form, which again shields us from that harmful UV light. This really allows aerobic species to really start taking off at that period. We see a lot of species actually now using that oxygen in the atmosphere, and we see kind of levels equaling out and becoming more suitable for life. So oxygen had to accumulate in the atmosphere before we see these aerobic bacteria and these aerobic species really flourishing. Now, after that, the oxygen allows our planet and it allows the species on our planet to become a lot more complex. So again, the oxygen allows the complexity of life to drastically increase about 2 billion years ago. This is where it starts to get a little bit confusing. Most of the plants that we see today are going to be these things called C3 plants. The reason why they're called C3 plants is one of those first molecules, that PGA. That's a 3-carbon molecule that we've named these plants after. You can see here it's carbon 3. It's that 3-carbon molecule. Part of the reason why we have a problem that's going to occur in plants with these C3 molecules, or C3, not molecules, I'm sorry, plants, is that during the day we're going to close the stomata usually, and that allows water to be conserved by the plant. Now, the problem here is CO2 can't enter if the plant stomata are closed. So yes, on one end, we're going to have water. It's going to be conserved and not leave the plant, but we can't intake carbon dioxide. It's like if you held your breath, you can't get oxygen in. That's going to be a problem for you, and it's going to be a much quicker problem for you than plants with CO2. But there will be a problem. The bigger problem is something called photorespiration. Photorespiration is when rubisco, remember that enzyme that fixed carbon dioxide into the 5-carbon RUBP? Rubisco can actually uptake oxygen as well. This is a big problem for a plant. You can see my little meme here. Rubisco likes oxygen more than it likes carbon dioxide. Now, when that happens, when there's high amounts of oxygen inside the plant versus carbon dioxide, that's a huge problem. We call that photorespiration. Whenever the plant intakes oxygen instead of carbon dioxide, it actually exhales CO2. Our whole goal for photosynthesis is to intake CO2 and to fix it into glucose. With photorespiration, so you can see this side here is photorespiration. You're exhaling CO2. In the Calvin cycle, you're intaking CO2 and making glucose. We don't do that when we when photorespiration occurs. We're not making glucose. We're exhaling CO2. That's a horrible thing for plants. So you're no longer just intaking CO2. You're stopping the intake of CO2 and you're respirating. You're exhaling carbon dioxide. That's bad for a plant. Now, when you might ask, like, oh, rubisco, that's an enzyme, and why can it have two different substrates? Well, again, it's one of those enzymes that does have multiple substrates. Very rare do those occur, but oddly enough, when photosynthesis was starting to evolve, oxygen levels were very low. So initially, rubisco didn't have to be selective for either CO2 or oxygen because there was almost no oxygen in the atmosphere. So because CO2 and oxygen are very small and they're very similar in structure, not exactly, but they are smaller and they do have similarities. Rubisco didn't need to be selective for either. It just needed to be selective for that type of molecule. So during that time, you had low amounts of oxygen. It didn't matter if it was selective or not. Now that it needs to be selective because there's higher amounts of oxygen in the atmosphere. So along comes these C4 plants. So C4 plants evolved a way around that photorespiration. The first molecule is going to be a 4-carbon molecule instead of a 3-carbon molecule. What C4 plants do is they're going to use this enzyme called Pep carboxylase instead of rubisco mainly. It still has rubisco in it, but the main enzyme we're going to use is this Pep carboxylase. It's also called PEPcase. So if you see PEPcase, just understand it's Pep carboxylase. Pep carboxylase doesn't use oxygen, so it can't use oxygen. So that means it can't go through photorespiration. Uses more energy, yes, but that means this can evolve, and these plants can live in hotter environments. You can see like corn, sugarcane, any organism or any species of plant that's really subjugated to really hot environments is going to be usually a C4 plant. Our next one here, and you can see the Pep carboxylase here in the C4 pathway. Instead of the first molecule being a 3-carbon, this oxalate is a 4-carbon molecule. The other type of plant that evolved around this problem of photorespiration are CAM plants. They usually open the stomata only during the night and uptake as much CO2 as they need during the night. And then they're going to, again, they also have a first molecule. It's 4 carbons long, but they regulate by just opening the stomata up during the night and closing them during the day. But they live in different type of environments. They live in usually hot and dry environments. So these are just two ways that plants have evolved ways around photorespiration.